Hello everyone, welcome back to the session. So in today's class we are going to discuss about the short answer question in the structural organization in animals. Okay, uh, first question will be describe the three types of cartilage. Okay, so basically in our body we are having three basic types of cartilage. We are going to see one by one. Okay, so the first question will be the hyaline cartilage. Okay, how the hyaline cartilage will appear. It will appear in the bluish white color. It is translucent. So we are having three basic terms, that is transparent and translucent and the opaque. Okay, transparent in the sense the light can pass through that medium. Okay, so that medium may be glass. Okay, so if the light passes through the glass, immediately the glass will easily pass all the light through the glass. Okay, so that is what a transparent. Translucent in the sense it is not completely transparent, it is not completely opaque, in the middle of this in the middle of the period. Okay, so the opaque in the sense it can't pass the light through the medium, so that is what opaque. So here the hyaline cartilage is the translucent and it is a glass like. Okay, so matrix, how it will be? It is homogeneous, it is similar throughout the cartilage with delicate collagen fibers. Which fibers is present in the hyaline cartilage? That is a collagen fibers. Okay, how the cartilage will be, what will be the strength? See, it is the weakest. Why? Because it is very flexible in nature. So the basic difference between bone and the cartilage, we can't bend the bone. Okay, but the cartilage in the sense it can bend. Okay, so it will be flexible when compared to the bone. Okay, so the most common type of cartilage, another important thing will be this. So we are having three basic types of cartilage. In that three types, the most common type of cartilage will be this hyaline cartilage, right? Now, perichondrium. What is perichondrium? See, this is present in this hyaline cartilage. Okay, perichondrium is present in the hyaline cartilage except in the articular cartilages. Okay, in the where it will be present. The location of this hyaline cartilage. Okay, so the embryonic endoskeleton of bony vertebrates. Okay, so the bony vertebrates, they are having a skeleton, more specifically the endoskeleton. See, exoskeleton and endoskeleton. We are having two different types of skeleton, exoskeleton and endoskeleton. Okay, so exoskeleton in the sense the skeleton which prevent the body, which protect the body by presenting outwards. Endoskeleton in the sense they are present inside the body and protecting them. Okay, so this is what the hyaline cartilage will be present in the embryonic endoskeleton of this bony vertebrates. Okay, so next, again the endoskeleton of cyclostomes. Cyclostomes in the sense those organisms which are not having jaws. Okay, without jaws. So that is what a cyclostomes. Cartilaginous fishes, and as the name called as a cartilaginous fishes, which is technically called as chondrichthyes. Okay, so that cartilaginous fishes, they are having a hyaline cartilage. Okay, now articular cartilages. Articular cartilages in the sense they are the joint surfaces of the long bones. See, consider this is a bone and this is the bone. These two bone has to join here. Okay, and that is called as a joint surfaces. And this hyaline cartilage is present in the articular cartilage. Articular cartilage in the sense this is present between the two bones as a joint surfaces. Now costal cartilages. Costal cartilages means sternal end of the ribs. So you could have come across with the word sternum and the ribs, and this costal cartilage is present in the sternal end of the ribs. Okay, so there we can find this hyaline cartilage. Now epiphyseal plates. So these were the plates. We can see the hyaline cartilage. Nasal septum, say the nose, tracheal rings, that is in the respiratory tract, bronchi, laryngeal cartilages. So you should know the respiratory tract, they are having the most common type of cartilage seen in the respiratory tract will be the hyaline cartilages. Now elastic cartilage. Elastic cartilage in the sense it is appeared in the yellowish due to the abundant elastic fibers. So you should know it will be in the yellow in color. Okay, yellow in color. Why? Because what is the name of the fibers present in the elastic cartilage? That will be the elastic fibers. what it contains an elastic fibers as well as the collagen fibers. See, in the hyaline cartilage we can see the collagen fibers, but in the elastic cartilage we are having the elastic fibers as well as the collagen fibers. What will be the properties? It provides the strength as well as the elasticity. That is why the name elastic fiber, elastic cartilages. Here the perichondrium will be present here. Okay, perichondrium is present in case of elastic cartilage. Now, where it is exactly present? Pinna of the external ear. So this is called as a pinna. We can see that cartilage present in the ears will be the elastic cartilage. Eustachian tubes, again is a part of a inner ear, and epiglottis. Epiglottis in the sense, see, we are having a respiratory tract and the digestive tract. Okay, so pharynx will be the common pathway of both water, both the food as well as the air. Okay, so after the pharynx, the air has to enter into the tracheae. Okay, pharynx, larynx. Okay, they have to enter into the larynx. In the digestive tract, the food has to be entered from the pharynx to the esophagus. Okay, so to prevent the pathway, the structure epiglottis is very essential. So this epiglottis can do its function properly, that is because of this elastic cartilages. Okay, and the third cartilage will be the fibrous cartilage. Okay, here they are having a bundles of the collagen fibers. The collagen fibers will be in the bundles. Okay, and the strength will be the stronger. So fibrous cartilage and the elastic cartilage and the hyaline cartilage. Okay, when compared to all these cartilages, fibrous cartilages is very strong. And perichondrium, here it will be absent. It is present in the elastic and hyaline, but it is not present in the fibrous cartilage. It is present between the intervertebral disc. See, this is an vertebral column. This is the center part, and here is also we can see the bone. Here also we can see the bone. Here also we can see the bone, and this is the bone. Okay, so this is the bone, this is the bone. Between these two bones we can see the intervertebral. That is what an intervertebral disc. So between these two bones we can see the fibrous cartilage. Pubic symphysis of the pelvic girdle. Okay, so here also we can see the fibrous cartilage. See, perichondrium in the sense, so this layer is called as a perichondrium. Okay, and the lacunae in the sense, this space is called as said to be as the lacunae, and here this space is called as the chondron. Okay, see, this is the chondrocytes. Okay, so this is the chondrocytes, and between the chondrocytes we can see the space that is called as a chondron. Inside the cell we are having the space that is called as a lacunae. Okay, so see, this is the lacunae and this is the matrix in the chondrocyte. We are taking one chondrocyte, so this is called as a chondrocyte. The nucleus is present in the chondrocytes, and this is the matrix, and the layer is called as a lacunae. Okay, now see, in the elastic fibers, again the layer is called as a perichondrium, and here it will be the lacunae, and this is the yellow fibers which is elastic in nature, and this is again a chondrocytes. Okay, next fibrous cartilage. In the fibrous cartilage again the chondrocytes will be there. Okay, and these are all the collagen fibers. Okay, so as it is rich in the collagen fibers, it appears white. Okay, so these are all the basic three types of cartilage. Okay, now the second question will be the Harvesian system. Okay, Harvesian system. So Harvesian system is also called as an osteon. Okay, Harvesian system is also called as an osteon. It is a structural and functional unit of the compact bone. So you are having the bone. See, the structural and functional unit of living organisms will be the cell. Okay, the structural and functional unit of kidney will be the nephrons. Okay, the structural and functional unit of the nervous system is neuron. Okay, so like in the same way, the structural and functional unit of a bone, compact bone, is called as the Harvesian system or osteon. Osteon is the structural and functional unit of the bone. So where it is present, where will be its location? It is found between the outer and inner circumferential lamellae of the compact bone, which means Bone, even though it is very strong, it is made up of a cells, no doubt in that. And between these two cells, between these two lamellae is nothing but a membrane. Between these two membrane, we are having a osteon. The space between the Harvesian systems are filled with the interstitial lamellae. So the space between the Harvesian systems or an osteon is called as an interstitial lamellae. Okay, how the Harvesian canal will be? Okay, runs parallel to the marrow cavity. See, consider this is a bone. This is a bone, and in the bone we are seeing this one soft structure which is called as a bone marrow. Okay, so in the bone marrow they are run parallelly. That canal system is called as the Harvesian canal. So what this canal is obviously consist of? See, this bone marrow is the site of synthesis of all the cells. Okay, so all the stem cells will be present here, bone marrow. So definitely they need an artery, veins and lymphatic vessels. So this Harvesian system runs parallel to the bone marrow cavity, as well as it contains the artery, veins and the lymphatic vessels. Okay, so the third one will be the concentric lamellae. Okay, so concentric lamellae in the sense it surrounds the Harvesian canal in the circular layers like this. This is what a concentric layer. Concentric lamellae. Okay, so in the circular layer, this Harvesian canal is surrounded by a lamellae which is called as a concentric lamellae, which is circular in nature. So the lacunae and the caniculi. So this lacunae and caniculi is very important. Lacunae is the sense small fluid filled spaces between the lamellae, each enclosing one osteocyte. See, this is an osteocyte. Osteocyte, and what is osteocyte and osteoblast? Osteocyte is considered as an inactive osteoblast. See, this is enclosing one osteocyte, which is the one circulating the osteocyte. The lamellae is covering an osteocytes. Okay, so they are the one lamellae and another lamellae. Between these they are having a spaces. In that spaces it will be filled with a fluid, and that fluid filled spaces is called as a lacunae. Okay, and what is caniculi? Caniculi in the sense the minute channels connecting the lacunae to each other and to the Harvesian canal. So this is the space, fluid filled space. We are technically call them as a lacunae. Here this lacunae, like in the sense all the lacunae is connecting together, connecting to each other with the Harvesian canal with the help of this caniculi. Right? Osteocyte possess the extend through the caniculi for the nutrient exchange. Definitely this osteocyte needs the nutrients. Even though it is inactive, it is not in the dead, it is in the live condition. Okay, so that's it needs a nutrients. Walkman's canals, it is a transverse or oblique canals that connect the Harvesian canals with each other, the peristomian and the marrow cavity. See, in the bone marrow, in the bone we are having a marrow through which it has been present in the marrow cavity, and the periosteum and the Harvesian canals. All these things has to be connected together with the help of a canal which is called as the Oltmann's canal. What is its function? Why it has to connect all these things together? Facilitate diffusion of nutrients and gases from the vascular supply. Okay, just to take nutrients to all these structures. So this is what a function of this Oltmann's canals. See, this is what a compact bone, how the bone will look like. Okay, so this structure it is called as the bone marrow. So the inner layer is called as an endosteum. Okay, so these layers with this membrane which is covering this bone marrow is called as the inner circumferential lamellae. So on the canal which is connecting this Harvesian canal to the bone marrow is called as the Oltmann's canal. Okay, so this is the Harvesian canal, and each one is surrounded. It is surrounding a lacunae that is called as a concentric lamellae, and one concentric lamellae. This is the another concentric lamellae. Between these two spaces is called as the interstitial lamellae. So this is the inner circumferential lamellae, and this is called as an outer circumferential lamellae, and entirely it will be covered by a membrane which is called as a periosteum. This is how the bone structure of bone will look like. Okay, The third question will be write short notes on the lymph. Okay, so there is a difference between blood and the lymph. What is the difference between blood and the lymph? Okay, blood without the RBC is called as a lymph. Just remember, so in the blood we are having an RBC, in the lymph we are not having an RBC. As RBC is not present in the lymph, it is little bit colorless fluid. Okay, but because of the presence of RBC in the blood, it will appear as a red in color. Okay, so the simple definition for the lymph will be the blood without the RBC. Okay, so the lymph is a colorless fluid that forms a part of the lymphatic system. So the lymph is nothing but a colorless fluid that forms the part that entirely forms the system that is called as a lymphatic system. So what is exactly present in the lymph? Okay, there will be no RBC will be present, no platelets, no plasma proteins. Okay, everything will be it lacks the RBC, platelets and large plasma proteins. Okay, so it rich in the leukocytes. So as it is present with the leukocytes is nothing but a WBC. Okay, so that is why this lymphatic system is very well involved in the immune system. Or why? Because it is present, the immune cells are present in the lymph. Okay, chiefly composed of the plasma and the lymphocytes. Okay, chiefly it is composed of the plasma as well as the lymphocytes. Even though all the other things is present, this is in the higher proportion which is present in the lymph. Okay, how we can compare this lymph with the tissue fluid? Comparison with the tissue fluid, so it contains the less nutrients and oxygens other than the tissue fluid. So lymph is somewhat different from other tissue fluid. So it contains less nutrients and oxygen when compared to the tissue fluid, but it contains more waters as well as the other metabolic waste. So the waste materials and the byproduct will be present in the lymph mode. How it is getting formed? It is formed in the interstitial spaces. So this is one cell and this is a cell, and the space between these two cells is called as a interstitial space. Okay, so due to the hydrostatic pressure at arteriolar ends, water, solutes and small proteins from the blood plasma filter into the interstitial spaces, forming the tissue fluid. Okay, so this is how, because of the pressure at the end of the arteriolar ends, there is a pressure created in the arteriole. So the water, solutes and small proteins from the blood plasma filter into the interstitial spaces. Okay, so as it has been entered, because of this pressure, all these water solutes from the cells that has been entered into the interstitial phases, we are interstitial spaces, we are calling them that particular fluid as a tissue fluid. Okay, what will be the return pathway? Most tissue fluid return to the blood capillaries. How it has been returned to the blood capillaries? Again, it will be the pressure. So here it will be the osmotic pressure at the venular ends. So this is nothing but an arterioles and this is nothing but a venules. Artery, the end branch of the artery is called as an arterioles, and the terminal branch of the veins is called as a venules. Okay, so the hydrostatic pressure, see, from the blood capillaries, it has been entered into the tissue fluid due to the hydrostatic pressure. Okay, from the interstitial fluid, it has to enter back to the blood capillaries through the pressure called the osmotic pressure. Now the small portion enters the lymphatic capillaries, forming the lymph. Okay, so the lymphatic capillaries together, we are calling them as a lymph. Okay, so another important thing, this lymphatic capillaries in the intestinal villi, we are calling them as the lacteals. Okay, for the absorption of the lipid. So how the transport route? What will be the transport route of the lymph? Okay, so the lymph flows through the lymphatic vessels and the ducts. Okay, the blood will be flowed through the blood vessels and blood capillaries. So like in the same way, this lymph will flows through the lymphatic vessels and the lymph ducts. Okay, so eventually this is drains into subclavian veins, return back to the bloodstream. Anyhow, it has to mix with the bloodstream, no doubt in that. Okay, now what is its function? It acts as an accessory route for returning the interstitial fluid to the blood. Okay, so from the interstitial fluid, it has to return back to the blood. Okay, so it may be acting as a supporting route for them. and it also helps in the immune defense, transport of fats and removal of waste. Okay, so transport of waste, which is nothing but if the lipid, if the fat molecules if it is getting digested, it has to be absorbed. Okay, so it has to be absorbed and transport to all the cells, all the parts of our body. So that transport of fats will be done taken in the lymph. Why this transport of fats will not happen in case of blood capillaries? Why? Because blood is a carrier of most of the nutrients. Okay, but why not the fats? Because the size of the fat is little bit higher, they cannot be enter easily through the blood capillaries. But before it has been entered into the blood capillaries, it has been carried by the lymph. Okay, so also, as because of the WBC is present in the lymph, it will be involving in the immune defenses. Okay, so that's all about the basic structure and importance of the lymph. Now the another question will be the structure of skeletal muscle. Okay, so basically we are having three different types of muscle. Okay, skeletal muscle, smooth muscle and cardiac muscle. Okay, so we are going to discuss about the skeletal muscle. So the basic thing will be this, whether it will be striated or non-striated. The skeletal muscle is striped, striated and voluntary. Okay, usually attaches to skeletal structure via the tendons. Okay, so see, muscle to bone and bone to bone. If the bone is getting attached with the bone, that is called as a tendons, and muscle to bone is called as a ligaments. Okay, so this is an important thing. So usually this skeletal structure, skeletal muscle is getting attached with the help of this tendons. And how the structure will be, how the membrane has been named in the skeletal muscles. Okay, so we are having an endomysium, perimysium and epimysium. The first thing will be the endomysium, thin connective tissue sheath surrounding each muscle fiber. So consider this is a muscle fiber. So this muscle fiber is getting surrounded by the sheath, and that is called as an endomysium. Okay, so perimysium in the sense connective tissue sheath surrounding the muscle, surrounding a bundle of the muscle fibers, which means also called as a fascicle. So this muscle fibers is called as a fascicle. Many muscle fibers we are calling them as a muscle bundle. So see, each muscle fiber, see, muscle is made up of the muscle bundle is made up of many muscle fibers. If it is surrounding each muscle fiber, we are calling them as an endomysium, which if it is surrounding the muscle bundle, we are calling them as the perimysium. Okay, so this is the structure of the skeletal muscle. Just look at this. In the skeletal muscle, we are having the membrane, that is a cell membrane of the muscle. We are calling them as the sarcolemma. Okay, and this band is called as a dark band, and this band is called as a light band, and definitely there will be the presence of this nucleus. Okay, so and the third structure will be, say we have studied about the endomysium, perimysium, now epimysium. Okay, so the epimysium, it is an outermost connective tissue sheath surrounding a group of fascicles to form a muscle. This is called as an epimysium. Okay, now the extensions. These layers may extend beyond the muscle to form cord-like tendon, sheet-like aponeurosis. So this is very, very, very important. Okay, these are all the structural modification or structural extensions of the skeletal muscle, right? Now, what are all the characteristic features of this muscle fibers? Okay, what will be the shape of the muscle fiber? It will be long, cylindrical and unbranched. Okay, the shape of the muscle fiber will be long, cylindrical and unbranched. And the nuclei here, it will be the multinucleated. There will be not a single uninucleated, it will be multinucleated, and all the nucleus will be in the oval shape. It is present in the peripheral cytoplasm. What is in the sense of this peripheral cytoplasm? If the nucleus is present in the center, it is called as a central nuclei. So if it is present in the periphery, that is called as a peripheral peripheral nuclei. Here the oval nuclei will be located in the peripheral cytoplasm. Okay, so now where it is getting originated? How it is getting originated? By fusion of cells, that is technically called as a syncytium. Okay, now what is sarcoplasm? See, sarcolemma, sarcoplasm, sarcoplasmic reticulum. Okay, so the plasma membrane present in the muscle cell is called as the sarcolemma. The cytoplasm present in the muscle cell is called as a sarcoplasm. The endoplasmic reticulum present in the muscle cell is called as a sarcoplasmic reticulum. Okay, so here it is a sarcoplasm. It contains many myofibrils showing the alternate dark and the light bands. Okay, so there will be an dark band is also present, light band is also present. So it looks like a partition or a septum. So we are technically call them as a striations. Okay, so functional traits. What will be the functional trait? Operates under what its function will be. It is voluntary in nature. First thing, it will be this: it has striations in nature. It has striations because of the presence of dark band and light band. The second important thing will be the voluntary in nature. So these are all the very important thing. Why? Because the smooth muscle and the cardiac muscle are involuntary in action. Okay, this is voluntary. What is voluntary and involuntary? Voluntary in the sense, if I want to move my hand like this, okay, under my conscious I am moving my hand. Okay, so this is what a voluntary. Involuntary in the sense, a blinking of the eyes. I am not voluntarily blinking my eyes, it is unconscious. Okay, it is my subconscious mind. So this is involuntary in action. So skeletal muscle, they are having voluntary in action. It contracts quickly and undergoes fatigue quickly. Okay, so it can contract their muscle quickly and it undergoes a fatigue also quickly. Okay, as it is voluntary in nature. Innervated by the somatic nervous system. So basically in the brain we are having CNS and PNS, central nervous system and peripheral nervous system. Okay, so in the central nervous system we are having the brain and the spinal cord. In the peripheral nervous system we are having a somatic nervous system as well as the autonomous nervous system. So this skeletal muscle voluntary action will be coming under the control of this somatic nervous system. Okay, so what about the regeneration process? How the muscle cell is getting regenerated? Because they are having a satellite cells. Satellite cells means quiescent stage, it will be mononucleate. Okay, these cells these satellite cells are quiescent stage, mononucleate and myogenic cells. Okay, so this helps in the limited regeneration of the skeletal muscle. So satellite cell is the one involving in the limited regeneration of the skeletal muscles. Okay, so this is what the structure of the skeletal muscles. Okay, now we are going to discuss about the structure of a cardiac muscle. Okay, how the cardiac muscle is look like. See, as I said before, cardiac muscle is involuntary in nature and this muscle is present only in the heart. It is found nowhere in our body in any of the other organ. Okay, so cardiac muscle will be present in the heart, that is involuntary in action. Just remember, if pumping of the heart is voluntary action, okay, we forget to pump the heart. Okay, so that is why nature has provided with such immense quality that involuntary in action. Okay, also this is also having the striations. Okay, so this is found in the myocardium of the heart. So in the heart also, in the skeletal muscle we have studied about this epimysium, perimysium and endomysium. So here it is called as a myocardium, endocardium and pericardium. Okay, so this cardiac muscle is present in the myocardium of the heart in the vertebrates. Okay, so this displays the sarcomeres similar to the skeletal muscle. This is also having the sarcomeres. How the cells is called as a myocardial cells, as it is muscle, that is myocardium means cardiac muscle, myocardial cells. And it will be short, cylindrical and branched. It will be mononucleate or binucleate. What if in case of skeletal muscle it will be multinucleate? So the ends of the cells forms a junction with the adjacent cells, which means what? This is the cell and this is the cells. They are forming a junction connecting with the adjacent cells. Okay, this is what the structure of the cardiac muscle, and the most important character will be the intercalated disc. Okay, so this is the cardiac muscle, and this is the nucleus, and this is the intercalated disc-like structure. As because of this presence of this intercalated disc, we are having the partitions, striations in the cardiac muscles. Okay, so what is intercellular connections? Okay, so as I have mentioned before, this is a cell and this is another cell. These two cytoplasm is getting connected with each other because of the junctions, and that is called as a gap junction. Okay, so because of this gap junction, the cells can be joined by the electrical synapse. It may be electrical synapse or through the gap junctions. Okay, so this allows the electrical impulse to conduct along the muscle fibers. So in the previous class, one of those classes, we have discussed about how the conduction of the heart, what will be the working principle of the conducting impulse of the heart. Okay, so there we have studied about the sinoatrial node, AV node, and bundle of his and Purkinje fibers, how they are conducting the impulse from one chamber to the another chamber, all because of this gap junction. Okay, so this electrical impulse, to carry the electrical impulse, gap junctions is very, very useful. Now another important thing will be the intercalated disc. Okay, so in the skeletal fibers, because of the presence of this dark band and a light band, it looks like a striations. Okay, so like in the same way, this intercalated disc, this is the dark lines throughout the cardiac muscle. We can see this intercalated disc. So this is the distinctive features of the cardiac muscle. This can be expected in the very short answer also. Okay, what will be the functional mechanism for this cardiac muscle? Cardiac muscle is highly resistant to fatigue. Okay, it is highly resistant to fatigue. Okay, but in case of skeletal muscle, it can be easily, it cannot be resistant to the fatigue. Okay, it is easily fall into the fatigue. It contains a numerous sarcosomes, okay, abundant myoglobins. Myoglobin in the sense oxygen storing pigment. In case of muscle, we are calling them as a myoglobin. If it is in the RBC, we are saying that as a hemoglobin. Copious blood supply. As it is rich in the myoglobin, it is rich in the oxygen supply and in the blood supply. These features support continuous aerobic respiration. So this is very important. So in the heart, we can experience a continuous oxygen supply. That is nothing but an aerobic respiration. Now that's all about the cardiac muscle. Now we are going to discuss about the structure of this multipolar neurons. Okay, so this multipolar neurons, neuron in the sense, this is called as the cell body. Okay, and this is the axon, and here we can see the myelin sheath. Okay, so here also we can see the myelin sheath. If the myelin sheath is present, we are calling them as the myelinated neuron. If the myelin sheath is absent in the axon, we are saying that as an unmyelinated neurons. Okay, so this is called as a dendrites. Okay, so we are having a cell body, one to many dendrites and single axon. This axon is nothing but this structure is called as an axon. Now cell body is also called as a perikaryon, cyton and soma, right? So it contains an abundant granular cytoplasm. So abundant granular cytoplasm and a large spherical nucleus. A large nucleus will be present here. The cytoplasm includes the Nissls bodies. Okay, so the Nissls bodies will be there. It represents the rough endoplasmic reticulum. So the Nissls bodies, this is homologous structure to the rough endoplasmic reticulum, where the ribosomes is present, that is involving in the protein synthesis. Okay, so Nissls bodies in the neuron, they are producing the protein. Now, so in case of a muscle, we are saying that as a muscle, many myofibrils together form the muscle bundle. Many muscle bundle, we are calling them as the muscle. So like in the same way, many neurofibrils together, we are calling them as a neuron. Okay, so lipofuscin granules are products of cellular wear and tear, accumulate with age. So lipofuscin granules, they are the product of cellular wear and tear. So cell wear and tear accumulate with age. So this is very important. This is somewhat related to the age factor. Now, see, just look at the structure of the neuron. This is a multipolar neuron. We are having a unipolar neuron and a bipolar neuron and the multipolar neuron. So one axon, many dendrites. This is called as the multipolar neuron. Okay, so the dendrites will be there, and the cytoplasm we can see the Nissen's granules. This structure is called as a cell body. In the cell body we can see the nucleus, and the cells which is present in the myelin sheath is called as a Schwann cell, and these are all this structure is called as an axon. Okay, so the sheath which is covering the axon, this is called as a myelin sheath, and one myelin sheath will be connected with another myelin sheath. This node structure is called as a nodes of Ranvier, and this terminal structure of the axon is called as an axon terminal. So in the axon terminal we are having a knob-like structure, vesicle-like structure, which is called as the synaptic knob. Okay, so now see in the cell body, see in case of the cell body is called as a perikaryon, cyton and soma. This is the another name for the cell body. So in the central nervous system we are having a nucleus. In the peripheral nervous system we are calling them as the ganglion, right? But everything is a cell body. Dendrites. Dendrites is a short branched process arising from the cell body. Also it contains the Nissen's granules as well as the neurofibrils. Okay, so this is very, very important. And another important point will be this. This dendrites is the one conduct the nerve impulse towards the cell body. Okay, towards the cell body afferent process. Okay, so this is very, very, very important. And now, so this is the one, see, so dendrites involving in the afferent process. Here it conducts the nerve impulses away from the cell body, that is afferent process. So the impulse is getting towards the carrying towards the cell body is done by the dendrites, that is an afferent process. Here from the cell body it is taken away. Okay, so that is an efferent process. It is going to reach the synapse where we can see the neuromuscular junction, where the neuron is getting touch with the meeting the muscle. Okay, it is a single long cylindrical process originated from the axon hillock of the cell body. And the membrane which is covering the axon is called as a axolemma, and inside the axolemma we can see the cytoplasm, we are calling them as an axoplasm. In that we can see the neurofibrils, and here there will be no Nissen's bodies. Nissen's bodies will be present inside the cytoplasm of the cell body. Okay, so it just the one gives rise to collateral branches, and it distally branches into telodendria. So this telodendria is called as an axon terminals. So in the terminal end we can see the synaptic knobs or the terminal buttons. Okay, the synaptic vesicle is the one, is the vesicle. Inside the vesicle they are having a numerous neurotransmitters. Here the neurotransmitters will be the acetylcholine. Okay, so now platelets, what will be the short nodes between the platelets and the synapse? Okay, so what will be the platelets? Okay, so platelets is also called as the thrombocytes. Basically in our blood we are having an RBC, WBC and the platelets. Okay, so this platelets is technically called as the thrombocytes. Okay, so this thrombocyte shape which is colorless, it has no nucleus. Very, very, very important. You should not think RBC is the one not having a nucleus, but besides that this platelets is also not having the nucleus. Okay, it is a round and oval biconvex disc. In case of RBC it will be biconcave, but here it will be biconvex like this. Okay, count it will be in the lakhs, 2,50,000 to 4,50,000. Okay, so it formed by the fragment of, so this mother cell of this platelet formation will be the megakaryocytes in the red bone marrow. What will be the lifespan? The lifespan of RBC will be 120 days. The lifespan of the platelets will be 5 to 9 days. Okay. What will be the function of these platelets? They are secreting the thromboplastin. It plays a very important role in the blood clotting. Okay. So the other damage to the capillary linings seal minor vascular openings. So if there will be any leakage of blood in the lining, so it will be go immediately there to seal them. Now what is synapse? Just now we have studied about the neuron. So with the axon branches, so this is an axon. Here there will be a thin fine filaments. This is what telodendria, we are calling them as the axon terminals. So in the terminal end we are having a knob-like structure. So this knob is called as a synaptic knobs or terminal buttons. In the synaptic knob, they are having the synaptic vesicles. This synaptic vesicles is filled with the neurotransmitters. Okay, so what will be the function of this synapse? So this axon terminals nerve impulse away from the cyton to the synapse. Okay, so from the cyton to the synapse, the nerve impulse has been transmitted with the help of this axon. Now, the synapse is the smallest gap between the stellodendria of one neuron. See, this is what a neuron. This is the axon terminals, and again it will be the another neuron. It will be nearer to the dendrites of the another neuron. So the the distance between the gap between the telodendria of one neuron and the dendrites of the next neuron. So this gaps is considered as a synapse. Okay. So here it is transferring the signal from one neuron to another neuron. So this gap is called as a neuromuscular communication. Okay, so in today's class we are going to discussed about this. We have finished the structural organization animals. Okay, so in tomorrow's class we'll see the next short answer question. Okay, thank you, bye.